It's a pleasure to meet you here.
Yeah, thanks for inviting me. It's my pleasure.
Thanks for taking the time to meet me here in Science Center.
So what are we doing today?
Let's head inside the Watson lab.
Okay, I'm excited to find out.
I see you have 12 badges. What are they?
Yeah, indeed I have 12 badges, but I see that you have many colorful badges as well, many of which I do not have.
My favorite are the Young Geneticist badge and the Marine Biologist badge. What are yours?
Exciting, those are the ones that I do not have. So I do not have a favorite badge, but I do have fond memories of many of them. For example, the astronomer, for example, the meteorologist. But in general, I like all the life sciences badges as well.
Did the Young Scientist projects inspire you through your career?
Absolutely. So I think the Young Scientist project is one that allows you to self-learn, although you get some supervisions from your teachers, but by and large you do it yourself. So those are the formative scientific experiments that I do, and that really inspire me for my career in science.
Okay. I prepared 2 experiments for us to do today, the gel electrophoresis and the wheat germ DNA extraction.
Looks really exciting. Shall we get cracking?
We should.
Hey Elizabeth, I'm curious, what is the soap for?
The soap is to break down the cell membrane so that we can extract the DNA from it.
Ah, I see. So why do we need the ethanol to be added inside?
So that it can precipitate the DNA so that we can see them.
Oh, that's the reason. So I see some white thread over here. What are these?
They are the DNA.
So those are the DNA that we want to extract. What is DNA, do you know?
It stands for deoxyribonucleic acid.
Wow, you definitely know your stuff. You're on track to be young geneticist.
Thank you. So can you tell me more about your work?
Yes, of course. So my work revolves around cell, proteins, and DNA. So, but you know, certain viruses do not have DNA. Instead, they have RNA, ribonucleic acids. So during the initial phase of COVID-19, we were called upon to set up and manage a COVID-19 diagnostic lab. So have you done a COVID-19 diagnostics before?
I don't understand what that is.
Have you done an ART before?
Yes, I did.
Ah, you have done an ART. So you know ART is based on antigen and proteins, right? Whereas the early phase of the pandemic, there wasn't ART. So we have to only, do the detections of COVID-19 based on nucleic acids. So that was the work which we had done. We harvested, we extracted the nucleic acids from patient samples, and then we run a PCR. Do you know what is a PCR?
I don't know what's a PCR.
Okay, so it's called polymerase chain reaction. It sort of amplify a lot of the nucleic acids, something like a photocopier. You know, you can copy a lot of the exact copy of that nucleic acids.
Oh.
Yeah. So now that we have extracted the DNA, shall we do something to maybe run a gel with it?
You mean gel electrophoresis?
Oh yes, gel electrophoresis. Is that the setup for it?
Yes.
Do you want to show me?
Okay.
Shall we get started?
Yes, I think we should. While we are waiting, why not I show you some of my projects?
Right, I'll be delighted to find out more.
This is task 9 about the pros and cons about having a clone.
Now it's about cloning. So what is the pro of having a clone?
When my friend invited me to a party, but my mom and dad says no, my clone will stay at home and I can go to a party.
Wow, that's so nice. What about the cons of having a clone?
So my clone will copy what I say.
Ah, okay, but make sure you share everything with your clones as well.
Okay.
What would be the other projects?
It's on about detached earlobe dominant and the attached earlobe recessive.
This is very interesting. I never really look at my earlobe. So what is my earlobe?
I think yours is detached earlobe.
Detached. I think I belong to the majority, like yourself. What else have you done for your Young Geneticist project?
It's on the extracting mango DNA one.
Alright, it seems like you've become a master of DNA extraction.
Yeah. Oh look, I think it's done.
Yes.
Let's take a look.
Right, so let me stop it and then we can take a look at the gel. Oh, this is very well run. You look at the length. So sample X. Which of the sample resembles that of the sample X?
I think it's sample D.
You're right, that's the one.
I think the smallest ones can travel the fastest. Is that correct?
You're right. So which is the dye that travels the fastest and the furthest?
That's, I think that B travels the fastest.
Okay, so it seems like the pink color dye. Alright. You know, in the actual gel electrophoresis with DNA, we could also stain those DNA with dye like ethidium bromide, and then we can visualize it using ultraviolet light. Yeah. Alright, this is very exciting. What is that?
This is something I prepared. I'm gonna give for you for the time.
Alright.
This is a DNA model keychain and the Young Geneticist badge.
I see. So this is a DNA model. So what are the letters over here?
A, which is adenine, pairs up with T, thymine. C, which is cytosine, binds with G, guanine.
Oh, that's very impressive. What about these orange colors?
The orange beads are the sugar phosphate backbone.
Very impressive. You made this yourself?
Yes.
Thank you so much. Oh, this is the badge for Young Geneticist. The one that you have printed over there. What is the figures over here?
I think it's DNA.
Okay. Wow, this is very nice. Maybe I'll pin it on. So, now I have 13 badges.
Yes.
Wow, thank you so much.
You're welcome. Bye-bye. Thank you so much. I have a question. Do you think, since my grandfather died from T-cell leukemia, do you think that the stem cell therapy can be used to treat this type of patients in the near future?
I'm sorry to hear that. Stem cell therapy is available in especially bone marrow transplant for blood cancer, but it's very difficult to find matching bone marrow. So in the future, stem cells could become any cell types, right? So we could actually use embryonic stem cell or induced pluripotent stem cell and then differentiate them to T cells for the treatment of patients. So that will be the future medicines and we should look forward to that.
Okay.